So what is the gut-brain axis? So this is my definition of the gut-brain axis. So, this is not Webster, but it's a bidirectional communication system that's important to the normal brain function. And the reason why this definition is important is for the longest time we thought about top-down control. We know our brain controls all our bodily systems and we use the autonomic nervous system and hormones, but what I'd like to focus on here is how the bottom-up works. So we know from both clinical and animal studies that the microbiota itself and other aspects of the gut directly influence brain function. And so it's not just the idea that your brain controls your body, but that your body also controls your brain. And the way it does this is, in fact, by using a lot of different systems. And so those are sort of included here just sort of as an overview. And each of these in itself could be a topic for conversation. But the reason I put these up here, so we have your neuroendocrine system, which is your hormones, your corticosterone. One of the big players in the gut-brain axis is stress. Stress modifies your gut system. There's no doubt that we all know that emotion relies to physiological feelings. Our gut feelings are just a reflection of the fact that this gut-brain axis is real in our everyday life. The other thing I'll emphasize is your gut-brain axis is part of normal, healthy life. It's part of the way we balance these systems that on a daily basis throw little targets at our homeostatic system. So the gut-brain axis is all-encompassing and actually is designed to put us back to balance. And so in disease states, any level of these different places that I've put on this slide, whether it be the hormones, whether it be the way we metabolize our food, whether it be the immune system, whether it be the way we connect our body and our brain through the autonomic nervous system or through the enteric nervous system, which not everybody knows about. It was interesting when Margot referred to the little brain. My little brain is the enteric nervous system that wraps your gastrointestinal tract and controls motility and things like that. But it also connects to your brain. And it has processes that are right next to your gut that are effectively sampling the inside lumen of your gut and is the first responder that can talk to the brain without your blood ever even getting a system. So your gut can talk to your brain through changes in the bacteria in your gut without an inflammatory response in your body, without a change in the short-chain fatty acids or other metabolites. So these systems are part of, integrated into how we live our everyday life. And so that's the importance of this slide. And then of course there's gut hormones. These can be produced by the gut themselves or by adipose tissue. So the whole sort of mood, food, metabolism can all come together in the gut-brain axis. And it's been around for a long time. So this just gives you a couple of examples, right back at Hippocrates, who suggested all disease begins in the gut. Or Eli Metchnikoff, who got the Nobel laureate for his work on the blood-brain barrier, but in fact wrote this book on the prolongation of life when he was older than most people, suggesting that it was the sour milk he drank every day of his life that kept him alive. And he in fact inspired the Japanese individual, Shirota, who developed the first yogurt with probiotics in it. So there's sort of this historical context that brilliant people in the past have known about the gut-brain axis and actually considered in this, the prolongation of life, the fact that it was the bacteria in the sour milk that was keeping him alive. So this idea is not new. And more recently, as everybody and their brother must know, this has been in the press, it's been in the news, and this just shows you the cover of everything from The Economist, the New York Times, and some of the top journals in science have covers related to microbiome, and these are all less than 2 years old. So this is just jumping off of the page, and if we look at the PubMed production, this idea of the number of papers that are being published in an area sort of reflects, the way the word, and this is just the word microbiota. So if you would, I think if you put gut flora in, you'd go back a little bit in history, but there's clear reasons why this emergence happens, and the primary reason is technology. And so prior to 10 years ago, all we could do is culture bacteria, and most of the bacteria in your gut or on your skin or in your other surfaces aren't culturable. So it wasn't until we got sequencing, which I'm sure you heard a lot about this morning from the genetics perspective. For the gut bacteria, it's the same thing. We can sequence them, but interestingly, they have 16S nucleic nucleic acids, which are distinct from humans. So if we take a fecal sample and we sequence it, we take other samples from the body and sequence the nucleic acids, we only get the bacterial signature. So that's a very useful tool if we want to map out what's going on. So let me tell you a little bit about your gut. Luckily, it's past lunch. So each of us contains about 100 trillion viable bacteria. That is 10 times the number of human cells you have in your body. So there's really more of them than there are of you. So this is a very important thing to remember, is that biology and evolution and all of the other things combined, we don't have these systems that are so robust if they're not important. So this is a very important system. There's more than 1,000 different species reflected, although each of us probably have 400 or 500 different species in our gut, which means there's a great heterogeneity across people and probably across disease types. So there's high interpersonal variability, but minimal intrapersonal variability. So that means that if I sequenced my gut bacteria today and I came back in 6 months and sequenced it again, it would look pretty much the same. Unless, of course, I took a course of antibiotics, or I changed my diet, or I suddenly introduced exercise. You know, over time, things change, and developmentally, there's some key times when the microbiome changes. Certainly the first year of life is important. As we transition through adolescence is important. And then when we start to age, about my age and older, you know, as inflammation increases across your body, your microbiota change. So these systems are all connected, but for the most part, in average adults, it's stable. And across populations, one of the things to remember, and there's a lot of work going on right now to think about what is the healthy microbiome, because before we can think about what's wrong with the microbiome or how we might use it diagnostically, we sort of have to understand what are the normal differences. So one of the things that has come out of that literature is that there are different, what they call enterotypes. So these are sort of groups of people who there's about 5 or 6 major phyla of bacteria that are in your gut, and some of them have, there's 2 of them that are predominant in most people, but there's also other groups of people that have a different profile. So there's sort of 2 or 3, maybe 4 healthy profiles that might be considered. What's interesting about that is that this might be a good tool if we're looking to look at what are the differences between a population to subtype them into groups that might be most susceptible to one treatment or to developing new treatments. Your microbiota is a really good timestamp of what you've been exposed to date in life, and some of these other things that affect it: genetics, age, diet, environmental factors. So it might be a useful tool if we were going to subdivide individuals into categories or subtypes. So more recently, studies have showed that gut-brain axis may be important to brain development, behavior, and mood. And in healthy people, I'll just stick on that for that, we've seen improved mood with probiotics, we've seen improved emotional centers with probiotics, and we've seen a benefit in anxiety and depressive measures and reduction in stress hormone levels in response to probiotics. So a probiotic, just to be clear, is a bacteria that's added into your system that has a health benefit, right? So we have commensals in our gut bacteria. Those are our everyday bacteria. They might be exactly the same species as a probiotic you find in the grocery store, but probiotics tend not to stay around. They're sort of like, you take them, they do their effect, and then they disappear. It doesn't change your own system permanently. So that's an important thing to remember about probiotics. But in one study out in California, they did a great study. So this is in healthy volunteers. And what they did is they gave healthy women fermented milk either with or without probiotics. And it's interesting because one of the systems that Margot was just talking about that's active in kids with autism when they were doing the task that included the insula. What this group did is they used an emotional face task and they did some baseline, but what they found is a network effect that the people that got the probiotics had a reduced activation of an emotional network that included some of these brain regions that are involved both in anxiety and in processing emotional stimuli, such as the insula, which we recently heard about. And so these systems were down-regulated after 28 days of probiotics. So this just demonstrates not that probiotics are end-all and be-all, but that we can, in fact, using a probiotic that doesn't necessarily change the long-term profile of our gut bacteria, influence brain centers. And there's a lot of work going on in both functional bowel disorders and in other systems that shows that probiotics affect the brain even sometimes before they affect the peripheral system. And one of the ways that the gut bacteria influences the brain, and I'll just put this up here because I believe it's a topic that people are interested in. We certainly in my lab have shown a lot about one of the ways that the microbiota talks to the brain is through the immune system. And so this picture here depicts some immune cells, and in fact, your adaptive immune system, which is your NK cells, your B lymphocytes, your T cells, or T lymphocytes, are all requiring your gut bacteria in order to develop. And so what we've demonstrated is this microbiota T cell interaction is very important in the anxiety phenotype that presents. And so mouse studies, clinical studies, all of the studies to date have definitely showed that if we reduce inflammation, which is one of the things we can do by changing the peripheral or the adaptive, the innate or the adaptive immune system, we infect anxiety. And that's just summarized here, that in all the different ways that we can manipulate gut bacteria from the germ-free mouse that has absolutely no bacteria at all. It's a little bit not clinically relevant, but nonetheless, the mouse in the bubble has low anxiety, whereas the mouse with chronic gut inflammation and high levels of cytokines and inflammatory mediators in their blood has the greatest anxiety. And we can go everywhere in between by gating both how much gut inflammation we have or how much peripheral inflammation we have, and we can change the level of anxiety. But what's interesting about this, in almost every one of these studies where they've given a probiotic, they have reduced the anxiety-like behavior. So there is no debate. We can walk away today with an understanding that microbiota are linked to anxiety-like behavior, and probiotics have a beneficial effect on the anxiety component of that in both mouse and clinical studies to date. So I'm just going to tell you a little bit about development. So importantly, exposure to microbes and colonization occurs at birth and continues throughout development. So unlike a lot of these earlier factors that we were talking about that had to do with fetal development, there's a little bit of microbiota action in utero, but really it's that trip down the vaginal tract that exposes us to our mother's vaginal bacteria that engages the whole process that starts the colonization. It takes babies about 1 year to develop a microbiota profile, and some things matter. So once they're there, they're important for host pathogen defense. Of course, our immune system is developing tolerance during this period. Allergy and susceptibility allergy is important happening during this window. Our microbiota also help us metabolize our food. And can produce themselves products that can act both locally in the gut or can go across the barrier in our bloodstream and communicate with other systems. And I have a picture later I'll show you that sort of summarizes some of that. And in healthy infants, there's a lot of dynamic changes, as I said, over that first year, but this is affected by a variety of things. Breastfed versus bottle-fed affects the profile of your microbiota. Vaginal versus C-section affects the profile of your microbiota. This doesn't say one is more healthy than the other. It says they're different. And some of the risk factors linked to immune disease could be linked to sort of trauma or to changes in mode of delivery. The breast milk story is coming pretty clean. There was a publication recently out. So what breast milk provides your baby with respect to microbiota are human milk oligosaccharides that tell your gut or tell the bacteria to there or favor the colonization of good bacteria in your gut. So it's the milk oligosaccharides that are produced in the breast milk that aid that. And what was recently determined that one of those milk oligosaccharides that is really good at making good bacteria is actually linked to the genetics of the mother. So, we are learning a little bit more about how it's a very dynamic environment, right? So the milk is good, and the reason why we don't get it from formula-fed is that we can't make that protein. So they believe me, the formula, Mead Johnson, I've been at a lot of their meetings, they can't make this human milk oligosaccharide synthetically. It only is made in the breast. So, and why it's important is if we take a look at the way different things develop. So on the top, we see the panel that shows it's a very immature microbiota. It sort of develops and it changes over time. Similarly, the next line is sort of a typical neuron developing, and this synaptic genesis and this dendritic arbor that builds connections, that connecting of the brain is happening at the same time as your gut is developing those microbiota. So if your microbiota to brain connection is involved in this, that's important that these developmental windows overlap. And on the bottom, this is just from a recent review out of John Cryan's group. It just shows that these important critical windows for brain development are and risk factors for when some different neurodevelopmental disorders present are overlapping. So this is an important system to consider when we think about mental health. I view the microbiota as a good representation of the environmental history of the individual. So when we start thinking about gene-environment interactions, looking at the microbiome or some of these factors of how the microbiome connects with your brain might be important, and they might include things like diet, stress, inflammation, and gut barrier integrity, which might be influenced by all of these other things. I'll just do a quick overview of some of the work that's been done in autism, and then I'll show you a picture that sort of overviews some of these mechanisms I've been talking about, and then we can answer some questions. So there's been a little bit of work in autism, less work in other neurodevelopmental disorders, although there has been one probiotic trial that has shown in mice that if you treat, in an animal model of OCD, if you treat them with probiotics, you eliminate the OCD features. So there's work starting up, I know there's studies going on in a lot of different centers. We don't yet have the data. One of the problems in this field is it's happening too fast. We were a small group, now we're a big group, and so we have to learn a little bit how to talk to each other. But there's definitely GI disturbances in children with autism. I'm not sure that this is there's people looking at how that's linked to the microbiota and whether or not gut therapy would help symptoms related to that. But as I just explained, this is sort of a normal, healthy communication system. I don't think it needs to get to the point of GI disturbances for the microbiota brain connection to be part of it. It could be smaller differences that are involved. The number of GI symptoms associates with severity. There's a couple of studies that have looked at specific bacteria, and what those show, in a nutshell, is that in situations where they see a difference in children with autism of the actual bacteria, they have more bacteria that are inflammatory in nature. They're part of the normal healthy commensals, but when you get more of them, potentially you're getting more of these toxic signals that could increase inflammation in a bigger way. So that's one of the theories of how this might be working. And there's at least one report that says short-term treatment with antibiotics can alleviate some symptoms. But I'm going to put up this picture here, which just talks about microbiota dysfunction. And what I showed you, if I can get the pointer up there, is it showing up there? No. No? No? It's on my screen. It's not on. Oops. Nonetheless, so dysfunction in the GI system can affect a lot of different areas, and some of these are important. It can affect your immune system, which can lead to higher levels of inflammation. Some of the things I haven't talked about is that it can affect the way that we're metabolizing our foods, and one of the things that it does is it metabolizes and produces short-chain fatty acids, which are products of sort of fiber and things in that domain, and those are very important to anti-inflammatory actions. It can also affect local production of serotonin, because most of the serotonin in your body is made in your gut. And what happens when there's some gut dysfunction is that your endochromaffin cells produce too much serotonin, and so you can get some changes in the balance of how your serotonin might go. And we actually know that if we stress, at least in rodent models, if we stress mice and change their microbiota, it actually affects their serotonergic system in the brain. So these changes we see locally, actually we can mirror in the animal studies, and we see similar changes in the brain. So I think I'll leave the information at that point and go ahead and take some questions if there are some.
Yes, I had a question in terms of when considering a probiotic, what type of bacteria like lactobacilli and the amounts of which, like 2 billion, what do you look for yourself personally when you're taking a probiotic? Let's say you've just had a round of,
Yeah,
you had a cold, you had some antibiotics, you want to reestablish your flora. What do you look at in terms of quality and quantity?
So I get this question every time I speak, so thank you. I should really put it on the first slide. And so I'll just preface it, I'm neither a physician or I have no qualifications to recommend a probiotic, but what I can tell you is there's not much difference in what's in the store. So they're almost all lactobacillus something or bifidobacteria. Those are the two sort of clinically demonstrated effective things. So if we give those to people, those are the ones that were used in those studies I told you that reduced mood or cortisol. So what's available on the market is pretty similar. The difference might be in the live versus dead. So in Canada, we can put probiotic on the label if there's bacteria in there. It doesn't mean the bacteria is alive. Those of us in the world think the live bacteria are a bit better than the dead bacteria, so some of the yogurts that have high levels sort of tend to even if we take the maximum dose that's out there, which is maybe 1 × 10⁹ bacteria a day or twice a day, it's a drop in the bucket compared to those 100 trillion in your gut, which is why they don't colonize. What they do is they talk to your local healthy bacteria and help them get their foot back in the system. So probiotics really act sort of not the way we expect. They don't stay around. You stop taking the probiotic, they're gone. When people have checked during probiotic treatment to see whether the bacteria has increased in number, it has. So they hang around every day while you're there, but 2 weeks after you stop taking them, that particular probiotic might not still be in your system. So I don't know if that completely answers your question, but they're all good that are out there as far as that. I would get them from if they're in the fridge, they're probably a good sign that they're got live bacteria in them. So if they need to be stored in your fridge, it's probably a better product than if you can just leave it on your shelf.
So when he gets sick and he has a fever, and it doesn't necessarily relate to gastro symptoms, but more like a flu or a pneumonia of some sort, even though he's getting less therapy, not attending school, during that period or shortly right after, we see huge developmental gains. I work at sick kids as a nurse in infectious diseases, so I started thinking, is there any studies out there that are looking at infection and its role in altering the mental processes of these kids long term after they've been ill and during those short bouts of
Yeah, I think Alana had forwarded me some of the questions, and yours was an interesting one. So there's no evidence that infection boosts any aspect of these systems, but there is evidence that in some kids, so when we look at immune deficiencies, some kids are over-immune active and some people are under-immune active. So if both of those, even when we look at the stress axis, right, if you have hyper stress reactivity or blunted stress reactivity, those can both be part of the disease process. So it's likely that, I can only speculate that, some system is being activated that's missing on the everyday basis. That doesn't mean that infection is the right approach. There's a gentleman down in the US who feels we should infect all babies because we don't get enough dirt in our developmental time window. So some of what happens in neurodevelopment is we need to see aspects of the immune system in order to wire those. And we know that those are important to wiring the brain. But I can't say that I have a mechanism in that particular case that I could point to.